class. Now we get to some mutation stuff. Okay, so here's the syllabus stuff. Describe how mutation and something we're going to talk about later here, so we'll skip that for now. Increased genetic variation. Well, if my recipe is this and I change it, which what mutation means, like maybe when I'm copying it, I just make a mistake and I put a C where an A should be. Well, now I've changed my recipe. It might be a big change, it might be a little change. In fact, they tell you they want you to know that a change in DNA, and the word mutation, sometimes it's used a little loosely, but its specific definition is a change in DNA that's then going to result in a change in RNA, that then is going to result in a change in a protein, that then might result in a different trait, right? Central dogma, right? That this change in DNA may not make any difference. Here's a new word, they snuck it in here, right? We'll get to that word in more detail in the next unit that we talk about. But what phenotype means is like trait. Brown hair is a phenotype. So we'll just define it as that right now, okay? So now they want you to logically be able to explain how a mutation in a gamete might result in phenotypic changes in an offspring. What's a gamete? I think we might have said this before. It's a sperm cell or an egg cell, right? If a mutation happens in one of those and they're on their way to combining to make you, right? Remember we called that a zygote. It's going to grow up to be you. Then that mutation is going to be in you. You're the offspring. It happened in the gamete, which is one of your parent cells, but it's the parent cell that ended up being part of making you. So now we'll look at a little more specifics about mutation. But this is as specific as they get. This is as specific as they get. They don't go to different types of mutations. We're going to do that a little bit. Okay. So sometimes a mutation happens. In fact, often a mutation happens that affects just one gene. We call that a gene mutation. And sometimes it's the smallest change that can possibly be. Just one base was changed. That kind of gene mutation is called a point mutation. Sometimes we're going to see that mutations can affect whole parts of chromosomes, a lot of genes. Remember we said this in our analogy, a chromosome's like a cookbook. It's got about 1,000 genes on it on the average. And a gene is each little section. Each little section, it might unzip and get transcribed to make a piece of RNA, to make a protein, right? That's what a gene is, one section of a chromosome. Sometimes changes, mutations happen that affect whole segments of chromosomes or sometimes entire chromosomes. And so those are simply called chromosome mutations. Not just one gene, but more than one gene, maybe an entire chromosome. Now we have these terms that kind of go back to what we just talked about. You have like two kinds of cells in your body, in one way to look at them. You have cells that are directly involved in reproduction. These are called germ cells, because what the word germ means is to give rise to. When we say a seed germinates, we means the little baby plant pops out of it. That's what germ means. It doesn't mean a thing that causes diseases. It means a thing that causes something. Literally, that's what it means. We've come to use it to mean things that cause disease. So these are the cells like egg cells and sperm cells. They are germ cells. But also the cells that divided to form them by that division that we call meiosis, that's in our next unit, those are also germ cells. They're directly involved in making new babies of all kinds, right? All of your other cells are called somatic cells. Soma means body, and even though these are part of your body, these germ cells, right? This is muscle cells, bone cells, brain cells. They're somatic cells. So here's the significance in terms of mutations. It's just what we talked about. This is little Johnny, right? Little Johnny stays out in the sun too long, and the ultraviolet radiation changes some of the bases in the DNA of some of his skin cells and causes them to divide too much. Johnny got skin cancer because of that mutation. Johnny's not going to pass that mutation on to his kids, because Johnny doesn't pass the DNA in his skin cells onto his kids. He passes the DNA in his germ cells onto his kids. If a mutation happens there, a germ mutation, then he will pass that on to his kids. So that's the difference between these kind of mutations in terms of what kind of cells do they happen in. Germ cells or somatic. Somatic mutations aren't passed down, germ mutations are. So how many do you think? I'll just let you ponder that for a while. Take a good look at the person sitting next to you. Okay, so now we're back to sickle cell and hemoglobin again. Remember, we already saw it as a classic example of this, how one change in the primary structure of a protein can produce a deadly disease, this sickle cell hemoglobin we talked about, right? Well, now that we understand that this protein gets put together based on these recipes that start with DNA being used to make RNA, being used to make protein, transcription, translation, right? Now we see that the change in the DNA that resulted in this one amino acid difference was just one base. They got changed by a copying error way back in history from a T to an A in a certain part. That resulted in a GAA codon, the result of transcription using that as a template, right, to a GUA, which says put valine here instead of what GAA says, put glutamine here. So this is, again, a point mutation. It's also a gene mutation. It affected just one gene. Specifically, it's the point kind of mutation. And this is a classic example of that. So how can mutations not result in change? Well, if you have, I think I have a diagram here. Good. Okay. Maybe not. Shoot. I'm going to have to draw over here. I deleted one. Okay. Let's say you have DNA that's CCC. So it codes for RNA that's GGG, right? And let's say you have a mutation where this C changes to an A. So now instead of GGG, the codon that gets put together on the messenger RNA is GGU. Well, I hope I'm right on this. If you look in the chart in GGG and GGU, they both say put the same amino acid here. So even though you had a mutation, it didn't affect the protein at all because the very same amino acid was put in the protein chain. This kind of mutation is called a silent mutation for that reason. And again, they want you to understand, without knowing that word, that this change in DNA doesn't necessarily change a trait because it doesn't necessarily change the protein. It changed the RNA a little bit, but because of the redundancy of the code, it may not cause a different protein to be put there. Here now are the kind of mutations that are the most significant by far. They're the kind when instead of what we had up here, which we call, I haven't said this yet, a base substitution, this base got substituted for that one, right? Here we have a base getting inserted or deleted. So here we have first a deletion. Compared to up here, we're comparing all of them to up here, right? This U right here got deleted. Now you see what that did. That shifted the reading frame. The reading frame used to be, starting at the start codon AUG, that established the reading frame. The next codon is AAA, the next one's UUU, then that. But now we have AUG, AAG, but now instead of UUU, because that U is out, now it's UUG. And that puts a different amino acid here. Phenylalanine was in there before, leucine is there now. And so every place after this deletion in this case because it has shifted the reading frame now, every amino acid after this might be different. It might not because of the way the code works, but it might be different every one after. This protein is definitely going to be quite different in its structure, therefore fold differently, therefore probably function differently. This kind of mutation is almost always the kind we call lethal. Depending on how important that protein is, this is likely going to cause a cell not to be able to function. So if we insert, right, instead of delete, we insert a base, and they chose right here, if we go back up here, they chose to insert a U right here, right after the start codon. Now this is going to change the reading frame also. Oops, messed up. This is going to change the reading frame. But in this case, in addition to that, what has happened here is it has created a stop codon way early. This protein is going to be really short, right? And so in this case, this, for no great reason, is called a nonsense mutation. I don't know why they chose that word. One where an early stop codon comes to be, and that could be because of a substitution, because of a insertion or like this, or a deletion like this. That could happen at any one of those, but this is also going to be a big change. So we kind of just went over what is a gene here. This is our best little description of it. Now we're going to have a few practice questions, which I'm going to skip and just do these in the in class. Okay, you can do all these, and I hopefully you can figure out the answer. And I am out. That was a long day. I'm going to jump in the ocean. Bye.